is your blood 0.2a, and this is going to be blood 0.2b. So we're going to talk about human blood types. And your blood type is going to be determined by the antigens that you have present on your red blood cells. And the antigens are like protein markers, or I sometimes think of them as name tags. And there are 3 possible antigens that you could have on your red blood cells, and they can combine to form 8 different possible blood types for humans. So the 3 antigens are A, B, and Rh. So you can see from the screen down here, the picture, that if you have type A blood, it means you make the A antigen. You have that marker on your red blood cells. If you have type B blood, you have the B antigen. If you have type AB blood, you have both the A and B antigens. What's important to note that people often confuse is that if you have type O blood, people want to say there's such a thing as an O antigen. There is not. So think of O means no. So what I mean by that is O means that you have no A or B antigens. So notice that the red blood cell is lacking either antigen when it's type O. This little picture doesn't take into account the Rh antigen. So this is only dealing with ABO blood groups, but there are also such a thing as having a positive or a negative blood type, and that's the Rh antigen that comes into play there. So if you have the Rh antigen, you're a positive blood type. If you don't have the Rh antigen, you're a negative blood type. When your body is exposed to foreign antigens, you form antibodies against them. We talked about that when we talked about lymphocytes. Those are like your memory cells. So the same thing holds true if you are given a foreign blood and it contains foreign antigens that are not on your red blood cells, then you're going to form antibodies against it. So then the next time you're exposed to that foreign blood, you're going to attack those foreign antigens with your antibodies, which is going to cause what we know as agglutination or blood clotting. This is obviously going to be a problem for you and can cause death. When you want to type someone's blood, like we're going to do in our lab, what you do is you put 3 drops of blood on a slide. These only show 2 because, again, they're not taking into account the Rh antigen, but we're going to do that. So you put 3 drops of blood on the slide, and then for each drop of blood, you're going to add a specific type of serum to it that contains one of the possible antibodies. So for the anti-A, well, anti-A's means that's the antibody A, so it's the lymphocyte that fights the A antigen. So if we place it on your drop of blood and agglutination or clotting occurs, then we know that you have the A antigen on your red blood cells. So we know that you have type A blood. If it also clots in the B well with the anti-B, then we know that you also have the B antigen, so now we know you're actually type AB. And we can just continue that process. You can see down here we don't see clotting in either the A or B wells, so we know you don't have either the A or B antigen, so you're type O. Again, we'll also add on the Rh well, and so if you see clotting, we know you're Rh positive. If you don't see clotting, you're Rh negative. This just goes through the different blood groups. We'll talk about this more in class, and we'll break down what are the most common blood types in the United States and what are the least common. This chart will deal with a lot. We want to make sure that you know all the 8 blood types, and you know what antigens each blood type possesses, and then you know what antibodies would be formed if they came in contact with a foreign blood type. And then we also will use that to figure out who can you donate blood to and who can you receive blood from for each of the 8 blood types. There's something called the universal recipient and the universal donor. The universal recipient happens to be AB positive blood. It is the type of blood that can receive blood from any other blood type. And the reason why is because it contains all 3 possible antigens, the A antigen, the B antigen, and the Rh. So then it doesn't form any antibodies against any of those antigens because it already has them. So it can receive from anyone. On the opposite end is the universal donor, which is O negative. That's the blood type that can donate to anyone. And the reason why is because O negative lacks all the antigens on its blood. So it'd be like giving you blood that doesn't have any protein markers on it. So there's no way your body can recognize it as being foreign and attack it. So that's the one that they're always begging for in the hospital so they can use them in the emergency situations. The last thing we're going to learn about is something you probably learned about in Biology 1 already, which is using genotypes and phenotypes so you can do Punnett squares and figure out what your possible blood types for your children are. So again, phenotype is a vocabulary term that you should know, and that is the expression of your trait. So that's the physical expression of it. In blood type, obviously you can't look at somebody and see their phenotype for their blood, but you can look at it on the blood slide when we do the lab, so we still consider that to be a phenotype. So again, there's those 8 possible phenotypes. Your genotype you can't see, it's your genetic code. It's the DNA that you got from your parents that codes for you to have a particular blood type. And on those alleles, you can have those little antigen codes that code for you to make either the A antigen, maybe the B antigen, the Rh antigen, or it might code for you to not make the antigen, which again would be type O blood. So if you're type A, there are 2 possible blood types or genotypes that you could have for your blood, depending on whether you're what we know as homozygous or whether you're heterozygous. Those are terms from Bio 1 again. Homo means same, hetero means different. So somebody who's homozygous type A means that they got 2 alleles that code for the same blood type. So for type A, they got 2 alleles, both coding for them to produce the A antigen. If they're heterozygous, they had one parent donate the allele for them to code in making the A antigen, and the other one gave them an allele that codes for them to make no antigens. And so you're going to make the A antigen. It wins the battle. So A and B are both dominant over the O allele. Homozygous type B is working the same way. It's just now you either have 2 parents coding for you to make the B antigen, or one parent codes for you to make the B antigen and the other one codes for you to make no antigens. So again, the B antigen wins out. So you have type B blood. Type AB is what we know as the codominant blood type because A and B are codominant. One doesn't dominate over the other. So if one parent codes for you to make type A antigens and the other one codes for you to make the B antigens, you're going to make both. So you're going to have type AB blood. Type O is the recessive blood type because the only way you can have it is for both parents to donate the allele that codes for you to not make any antigens. You don't make the A or the B. So let's equate that into how we would do a Punnett square. If you had the problem where you were to cross a heterozygous type A with a heterozygous type B and then determine their possible genotypes and phenotypes, you would do the following. For a heterozygous type A, remember they have one allele that has the A antigen on it and the other allele that's lacking an antigen. For the type B heterozygous, same thing. They have one coding for a B antigen, the other one coding for no antigens. So then remember you bring down and across to fill out your Punnett square. So you bring this one down and this one across. So you finish filling out your Punnett square. The genotypes are what you actually see in the squares. Those are the actual genotypes, the genetic codes that would be in their blood, in their DNA. And then the phenotypes is what would they actually have for a blood type. So this one equates to type AB blood. This one equates to B because the B wins out. This one equates to type A blood for the phenotype. And finally, this one equates to type O. You have now completed your Blood.2 video lectures, A and B.